Hi there. In this video, I'll be talking about DNA repair, and it sort of is an addition to the DNA mutation video. Because the DNA mutates so often, then to have large genomes, and I'm speaking about genomes the size of bacteria and eukaryotes and archaea, DNA repair systems have evolved. So there are some major ones, that is base excision repair, mismatch repair, nucleotide excision repair, and recombination repair. And I'll talk about each one of these. But essentially, base excision repair is stimulated when there's an inappropriate base or a base that is missing from the backbone. And the first thing that happens is there's removal of the strand with the mutation, and then DNA polymerization and ligation fills in the gap. And sometimes this gap can be over 100 nucleotides long. So it doesn't simply take out the lesion, but it takes out a long stretch of DNA. Mismatch repair is when there are mismatched nucleotides, like an AC pair above. And again, there are enzymes that clip out the offending strand, although sometimes it's unclear which one is the offending strand, so sometimes the other strand is removed. And then there is DNA polymerization and ligation to fill in the correct base, as well as a large number of other surrounding nucleotides. Nucleotide excision repair is primarily when a major error is detected, such as a pyrimidine dimer, where no pairing occurs and there's usually a bulge at that region. And so the first thing that's done is several different proteins surround that area, clip out the DNA, and then fill in the correct nucleotide sequence based on the template strand that's left over. Recombination and recombination repair are a little more extensive, and I'll talk about those towards the end of this video. In addition to the repairs I've already discussed, mismatch repair, nucleotide excision repair, base excision repair, there are also things that cause direct reversal of the mutation, and in that case, they actually just exchange the base that's the mutated base. So here is base excision repair. This will be something like an AU pair caused by a mutation of a methyl C. In this case, the damaged base is recognized, and there's a specific DNA glycosylase that moves in there and cuts the damaged base out. And then there's an AP endonuclease, that is an apurinic apyrimidinic, because there's no base in that site. It clips out the backbone surrounding that lesion. And then DNA polymerase comes in and starts filling in at that nicked region, filling in much more than just the nicked region. And then DNA ligase seals the last two nucleotides together in the backbone. Nucleotide excision repair is signaled when there's usually a bulge or a distorted region in the DNA. There are constantly DNA binding proteins that move along the DNA to find lesions in the DNA. And so in this case, several nucleotides are chopped out, including the thymidine dimer, or thymine dimer, and then DNA polymerase repairs that single-stranded region, and DNA ligase seals up the last two nucleotides together. Although mismatch repair and nucleotide excision repair on the surface seem similar, they require different proteins and different enzymes, and so they're very different processes. One disease that I mentioned in the DNA mutation video is xeroderma pigmentosum. And these are mutations in this pathway, in the nucleotide excision repair pathway. So if these people with these mutations go out in the sun, they immediately get lesions from that, in addition to sunburn, but lesions from that. And some of them can turn into skin cancers. This bottom picture here is a girl who is happy enough to go outside because she has a UV shield, so they can go out for a short amount of time outside, but not for a long period of time. Also, they have to avoid some types of fluorescent lighting, which also emits some UV irradiation. When I see people with this disease, I think that each time you go out into the sun, you're also damaging your DNA. But your repair systems patch up most of those errors. And although you can get this disease by spontaneous mutation of some of these proteins, it is mainly inherited from parents. So here's that nucleotide excision repair pathway. If you have UV damage up at the top, then several proteins run along the DNA and recognize that, and then it recruits other proteins into the same area, including an endonuclease, which clips out the fragment of DNA that's in error. And then you have DNA polymerase and ligation synthesizing a new strand. If you note that some of these proteins listed here are XP, 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 that stands for xeroderma pigmentosum, so those are some of the proteins that are mutated that cause this nucleotide excision repair to fail in the people with xeroderma pigmentosum. So here's a summary of some of those repair mechanisms. There's direct reversal, changing of a base at a time, mismatch repair. There's nucleotide excision repair, base excision repair, homologous recombination, which I'll talk about in a little bit, and then non-homologous end joining, which is sort of an inefficient process. And then there are photolysases in bacteria, plants, archaea, fungi, and some animals where there's direct reversal of pyrimidine dimers, so you don't need the nucleotide excision repair system, but they can rely on a different system, which incidentally is stimulated by light, so it's most active when it's very sunny out. Now I want to talk about genetic recombination, which is both a repair system for getting rid of lesions. As you know, that's its main purpose in meiosis, to engage in crossovers which will join together the best genes and get rid of the deleterious genes, which doesn't always work, but this is one way that this happens. So you can have, mainly people think over here in meiosis, you get crossing over between sister chromatids as well as homologous chromosomes. During mitosis, there also are exchanges, although people don't realize that those are going on as often. And then in diploids, there are often crossovers between non-homologous chromosomes, and many of these cause tumors and cancers or lethal conditions. So a crossover can occur between two double-stranded DNA molecules. In this case, I have two different DNA molecules, one with dominant alleles on it and one with all recessive alleles, just to illustrate the point. They could be anything. The initial event is a single-strand break, which causes a nick in the DNA, and then that recruits single-stranded binding proteins to that area so that the DNA can denature into the two separate strands. So you can think of it as those DNA binding proteins, the single-stranded binding proteins, binding to the hydrophobic bases so that they can be pulled away from that hydrophobic center of the DNA. Next, there is strand invasion, so this red strand starts displacing the blue strand on that dominant allele DNA. And that causes this looped out region in the blue chromosome, which is called a D-loop, because you can actually see those in an electron microscope and they look like the letter D. Next, that blue strand, because it's complementary to the red strand on the other side, starts binding to that, and now you have a crossover, but it's not quite resolved yet. But you can already see that at the crossover point here, there's two different regions where there's what's called a heteroduplex, that is, the strand from one of the original chromosomes, and the opposite strand is from the other chromosome. So if those were dominant and recessive alleles, now you have sort of a 50-50 mix of the two on that double-stranded DNA. And then finally, there's ligation on the two strands over here, and there's a definite crossover that's still unresolved on the other side. So this results in this structure here, where you have the dominant A allele, the dominant D allele on one side of this, and then a heteroduplex region in the middle, and then on the other side you have the recessive allele of A, the recessive allele of D, and then a heteroduplex region in the middle of that molecule, that part of the molecule. And this can be resolved in two different ways. So here's the first way. The crossover is stretched out, and then it's cut and ligated together. And this creates the two chromosomes that look like this. One has the dominant allele A here and the dominant allele D there, so it really hasn't changed the ends of that chromosome. But the middle part is a heteroduplex containing partly the dominant allele and partly the recessive allele. And the opposite is true of the other chromosome, which has the recessive alleles on both ends and then the heteroduplex region in the middle. There is a second way to resolve the crossover, and that's first to rotate this bottom part around. So you reverse the A and the B sides, and it opens up this middle, often called a chi structure. Then rotate these two here, exchanging the dominant D with the recessive A. You really haven't changed what's connected with what at this point. And you might think, well, these are a lot of contortions to make with this DNA, but they actually occur. These contortions of the DNA occur in nature all the time. And this time you cut across here and then resolve those two chromosomes. And now here they are, and now the chromosomes have fundamentally changed, so that this chromosome here has the dominant A allele on one end, but the recessive D allele on the other end, and still this heteroduplex region. And the opposite is true of this chromosome. It has the recessive A combined with the dominant D, and then the heteroduplex region in the middle. So the part of the process that is the repair process rather than the recombinatorial process is you can exchange chromosome arms to get rid of deleterious alleles. But in the middle, you can also generate new types of genes due to that heteroduplex region. So that middle portion will use either the one strand, the blue strand, let's call it, or it'll use the red strand to correct any errors in that. So that's how it's also a repair system. In some bacteria that are highly resistant to radioactivity that would kill you or me, they have recombination systems that will bring together broken pieces of DNA to reconstruct the genome. So this happens in Deinococcus radiodurans that essentially its genome, its chromosome can be blasted apart by the radioactivity. But the recombination systems are so much better than ours. It can actually reconstruct by recombination the entire genome. So next I want to talk just a little bit about more about recombination and its importance to organisms and evolution. So as I showed a little while ago, recombination occurs much more often than people realize. Most people think of recombination only in meiosis to remix the different alleles for the gametes, but that's not the only place it happens. It does occur during mitosis very often, actually, even in human cells, especially in highly repetitive regions like the ribosomal RNA region. It's very active in those regions. But it also can recombine among non-homologous chromosomes Often this is deleterious, but it also leads to speciation events where you have now new types of chromosomes rather than the original set. So here's the normal condition that you usually see during meiosis, where you have a pair of chromosomes, homologous chromosomes, that then duplicate, and then during meiosis you get crossovers exchanging different arms of those chromosomes, and then during the second division of meiosis, those chromosomes with now different combinations of alleles separate and become the nucleic acids in the gametes. However, you can get crossovers that cause other major changes within the chromosome. So you can have deletions and insertions and inversions of chromosome regions. Often those will then lead to a deletion of certain functions in the cell, or duplication sometimes leads to deleterious or advantageous traits as well. In some cases, you can get rather large insertions of pieces of chromosomes due to recombination events usually. So here's a piece of chromosome number 4 being inserted into chromosome number 20, which does cause a type of cancer in humans. Also, here's a translocation where there's been a single crossover between chromosome 4 and chromosome 20, leading to this situation where now you have two different aberrant chromosomes, and that also leads to a type of cancer. And then this one up here, crossover between chromosome 12 and chromosome 17, causes two new types of chromosomes here, and that causes a certain type of leukemia. Also, there's a crossover between chromosome 14 and 21 that causes a type of Down syndrome. Normally people think of Down syndrome of being trisomy of 21. In this case, the individuals are diploid, but part of the chromosome 21 has been exchanged with a different chromosome, and that also causes Down syndrome symptoms. And here's another example of a translocation between chromosome 8 and chromosome 14 in humans. Here you have chromosome 8 getting a little shorter by adding a piece of chromosome 14 and losing this piece of it, which makes chromosome 14 a little longer, where it has gained a piece of chromosome 8. And this causes Burkitt's lymphoma, which is a tumor of B lymphocytes of the immune system. And this is just a general diagram of some of the translocations that cause diseases. So up here, a crossover, a translocation between chromosome 2 and 5 causes a large cell lymphoma. Here's a crossover between chromosome 1 and chromosome 12 that causes AML, a type of leukemia. Chromosome 1 and chromosome 11, this translocation can cause some types of schizophrenia. Down here, the one I mentioned between chromosome 21 and 14 causes a type of Down syndrome. And there are others up here that cause mainly cancers of lymphomas and leukemias, blood cell cancers, because they often have recombination events normally going on in those cells, but occasionally they go wrong and create a translocation and a cancer or other disease. In many cases, these diseases are caused by a fusion gene, which causes a fusion transcript and a fusion protein. So two genes on those chromosomes, on different chromosomes, you get a crossover. Now you have a new type of gene, which on one end has the exons for one of the proteins, and the other end has exons for the other end of the protein. And so when you get that fusion transcript and fusion protein, often this can increase cell proliferation, decrease apoptosis or cell death, decrease differentiation of those cells, and also increase motility, making them more likely to metastasize. These gene fusion events are of a couple of type. The first is expression of a fusion gene as a fusion protein in the end, which has a different function than the original proteins had. The second one is one where a new promoter is placed at the front of a particular gene so that it has it increases in expression or concentration in the cell. So both of those things can happen. And occasionally when this happens, either of these two situations, then a tumor cell is initiated. Although these recombination events cause diseases, including cancer, they're a part of evolutionary processes, so that without those processes, then evolution would not have occurred, diploids wouldn't have occurred, large genomes would not be possible. So it's really an unwanted effect that they cause, also diseases and cancers, but they've really been a big driver of evolution. So here are a few examples of how they have caused advances in evolution. If you look at Trypanosome brucei, it has a surface on its flagella where there's a protein sticking out, and that's what the immune system of the host organism reacts with. So that when it first infects, the immune system then gets to work on that particular protein that's on its flagellum and attacks it and tries to kill it. However, within that gene, it has several variable regions, and the chromosomes can cross over in those variable regions, creating new versions of those proteins. So the different individuals have different mutations, different crossovers, and they will produce different proteins on their flagella. And so that now the immune system doesn't recognize the newly formed proteins. It kills all of the organisms with the old proteins, but now you can get further growth of these trypanosomes because now the immune system doesn't recognize them. So it has several mechanisms for evading the immune system and undergoing recombination of one sort or another. The first is gene conversion. So here is the expressed version up here, and then here are the extra copies of parts of that protein. And by recombination, it can flip one of those versions out and flip a new version in to change that surface protein. In another mechanism, it has two regions on those different chromosomes, and one part is expressed and the other part is not expressed, and it can exchange those pieces so that the unexpressed version can be moved to the location where it then is expressed and then create a new type of protein, surface protein in that case. And then finally, without recombination, it can switch transcription from one chromosome to the other chromosome, so that at one time it will produce one type of protein, and at a different time it can produce a different type of protein, and in that way evade the immune system. Incidentally, this coronavirus recently has been in the human immune in humans for a while because it has developed a system for evading the human immune system also, at least for a time. Speaking of the human immune system, it's a product of recombination also. So immunoglobulins, or antibodies, are proteins that have constant regions that interact with the host cells, and then variable regions out here towards the end that interact with the antigens, or the proteins that are formed by the viruses or any invading organism. And there are several different types of these, including IgG, IgM, and other types. So when you think of it, how can an immune system recognize so many different proteins, so many different antigens formed by so many different organisms? And the reason is that recombination can recombine different areas of these immunoglobulin genes in order to find a combination that works in interacting with the foreign antigen. So in the genome, there are V segments, which are variable segments. And then there are D and J elements for joining pieces together. And then there are constant regions here that interact with the host cell. So the first thing that happens is you get recombination in that organism between the D and J segments, and then further recombination to join 3 parts of the V region to the D and J regions. And so by the time you get this far, the cells have lost all of the other parts of those genes. So they only will focus on producing one type of antibody at a time. And then they're mature enough, or they're mature by that time, so they can't form any other type of antibody. That's the only type they'll ever form once they're mature. So here's another view of the same process. There are 150 genes in 15 families in the V region, so a lot in the germline. And then 12D for J and 8 constant regions. And by the time you get recombination, you reduce the number of some of the D and J, and then final recombination reduces it to only a few versions of the V region, so that each of the B cell produce only one type of antibody for each cell. And that's all that they can produce and present at their cell surface. Because of the diversity in the V region, then, they can produce literally billions upon billions of different combinations of antibodies. And notice that each one of the immunoglobulins has a heavy chain, a longer chain, and a lighter chain. So each of these come from separate loci within the genome of those organisms. So you have variable regions for each, and then D regions for this one, J regions for both, and then C regions for both. And then those fit together in the final combinations of these antibodies. So again, literally billions of different antibodies can be produced by these cells. But as they mature, they produce only one type for a specific antigen. So just to bring this back to DNA repair, recombination, while it is important in DNA repair, it's also evolved many different functions, including the removal of deleterious mutations, the collection of advantageous mutations and alleles, as well as ways to evade immune systems, ways to create new proteins, new enzymes, and in some cases it's really speeded up evolution, because now you can have crossovers to create new versions of proteins. Okay, so that's all I have to say about DNA repair, and I'll talk to you later.